everyone, Gayatri Prem Kumar here, and today we are going to discuss about the next slide preparation, that is study of mitosis in onion root tip. And I have given the pollen tube germination link in the description. You can also have a look at that video. And this video we are going to discuss about the introduction. What is the introduction or the concept that is behind the mitosis in onion root tip, as well as the aim, materials required, procedure to be done, and what is the observation. Finally, what do you observe at the end of the experiment? And there comes the precautions to be followed by the experiment when you're doing the experiment in the practical point of view, and what are the possible viva questions that will be asked. Understanding of what is mitosis and how it is different from the meiosis. The mitosis, we all know that mitosis is an equational division, equal division. It means one cell divides to form two daughter cells which have the equal number of chromosomes, and that forms in the somatic or the body cell. The next one comes the meiosis. So when we talk about the meiosis, it is a reductional division where the number of chromosomes is reduced to half the number, and two such division takes place, meiosis 1 and meiosis 2, and there will be four daughter cells are formed with half the number of chromosomes, and it takes place in the sex cells. So this is the basic difference about the mitosis and meiosis. And then coming back to the onion root tip. So itself, it is a basic cell division. So what is the cell division? So what are two different kind of division we have? One is the cytoplasmic division and the other one is the nuclear division. So again, when I'm talking about the cytoplasmic division, where the cytoplasm divides into two, nuclear division is called as karyokinesis, where it involves many different steps, and that is the division of nucleus, and cytokinesis for the division of cytoplasm. Again, in the division of nucleus, we have different steps because it is not a single step process. It is a lot of many different steps that is happening. And what are the different steps we have in this karyokinesis? That include the prophase, metaphase, anaphase, and telophase, and in each phases we can see the nuclear membrane dissolves, and then the spindle fibers separate, and how the chromatin thread again it untangles. So all these things will be like different steps that is given about each phases that is happening in the karyokinesis. So this is the basic introduction about this experiment. So what you're going to study about, and cut the tip of the root of the onion, which is grown either in the water or in the soil, and that we have to employ different kind of treatment procedure and sustained procedure and observe under the microscope the different stages of mitosis. This is the aim of the experiment. This is going to be your aim, but you're not going to write that much lengthy. You're going to write it in a very crisp and short way to study various stages of mitosis by preparing acetocarmine stain. Why we are mentioning acetocarmine, not the stained amount, we are mentioning specially acetocarmine stain. The reason is acetocarmine is a stain that we are going to use it over here. Why not saffron or any other stain? The reason is acetocarmine used to stain the nucleus of the cell, and that is why we are using acetocarmine stained mount of the onion root tip. So this is going to be the aim of the experiment. And how we are going to keep the onion? Maybe either you take a conical flask or a bottled one. The base of the onion should be dipped completely in the water. That way, the root to form, and the root will be one which is immersed in the water, white slender root. And sometimes if you leave it longer, you can see the green shoot that develops on the top of the onion. And then what you do is you take the onion and trim only tip of the onion, not the other part. You take only the tip of the onion. Why you take only the tip of the onion? Because actively dividing cells are present only in the tip of the onion. So you can cut is taken, and then you follow the other procedure. If the question is asked why you take the tip of the root, because actively dividing cells are present, the materials required for doing this experiment. So obviously it is about the onion root tip, and we do require onion bulbs. In this way we have to keep it for 1 week for the day of the practical examination. So you have to keep the onion ready prior 1 week or 10 days so that the onion bulbs are ready for the experiment to be done. And then it can be done either with a conical flask. You can immerse it in the conical flask, or any other glass bottles can be taken. And then the petri dish for the washing procedure, to cut the root tip and place it in the fixatives. For all these purposes, we require petri dish, scissors to cut the tip of the root, and then the forceps to take the root tip from one solution to the other solution, and then the teasing needles are required. just to press, immerse it in the stain, or rinse it with water. And then ethanol and acetic acid combination as fixative in the ratio. And then we have used one normal hydrochloric acid and give a gentle warm heating at 60 degrees Celsius. And then we use the stain acetocarbon, which I discussed already, which is there in the aim. And then we use distilled water for the washing purpose. And microscope, we use microscope for visualizing different stages of mitosis. And excess stain can be removed using blotting paper, slides to keep the root tip, and then the cover slip to place it on top of that. Now coming back to the procedure of this experiment. So already if you take any onion, you can see the roots are old. So first, before you're keeping, before in the water, you have to trim the old roots that are present in the onion. Just trim it, and then you immerse it in the water prior 1 week of the experiment. Whether it is the normal practical or the board practical, you have to keep the onions ready before 1 week. And then you have to cut. After 1 week, you can see the white slender root has grown. So you have to cut only the tip of the root. Again and again I'm insisting, cut only the tip of the root. 5 mm is enough, because that is the region you can see the actively dividing region, actively dividing cells. And then you just place it in a solution, a combination of 1:3 acetic acid and ethanol. So this fixative agent, acetic acid and ethanol, the ratio of 1:3 is taken. For example, like you have 1 ml of acetic acid means 3 ml of ethanol. So in this proportion, you just take the fixative and place it in that for 1 hour. Your time accordingly. So this takes 1 hour. Suppose this experiment comes for your practical, what you can do? You can keep the fixative prior, you can keep it for 1 hour. So by the time you do your writing work, the fixative will be ready. So this is how you have to manage your time. And about this, I have given the time management, how to manage your time during the practicals, and other tips I have given. And that link will also be shared in the description. You can have a look at that. And then coming back to the warming. So after this 1 hour, you have to take that and place it in a slide or again a vial where you can HCl. You can take some quantity of one normal HCl and warm it to a temperature of 60 degrees and not more than that, by gentle warming, and leave it for 15 minutes. So what is the purpose of adding these acids and these kind of heat treatment? To break the cell open, and thereby the cells remain separate, and when you're observing under the microscope. And then you have to wash it with, after this acid treatment, you have to wash it with water. And then all this procedure is over, you have to place a drop of stain acetocarbon, and this root tip what you have subjected to this acetic acid ethanol treatment and HCl treatment, and then you just place it in the slide containing acetocarbon, and then gently place the cover slip without air bubble. Press or squash it again gently, mild heating to make it remain in that place. And then you have to observe under the microscope to view the various stages of mitosis of this onion roots. So what you will observe under the microscope when you're observing? So we have seen the materials required, procedure. The next one is the observation. So what you will observe under the microscope when you're observing? You will observe the different stages. I have mentioned the initial stage, no, like prophase, metaphase, anaphase. So that different stages you will be observing under the microscope. And till whichever stage, maybe in your record you will be writing all the stages. Till the examination point, whichever stage you're observing, it could be prophase, metaphase. So whatever stage, like what you observe that stage, so that stage you have to write, and that how the chromatin, spindle fiber, nuclear membrane, nucleolus, how they appear, disappear, all these things you will mention about the description what you have written in your record. And following that, you will be drawing the diagram also. And that will remind your observation for this experiment. And then coming to the precautions, what kind of precaution that has to be followed while doing this experiment? The very important thing is that the time of taking the root tip. So you cannot cut the root tip at any of the time. There is a timing. In case of summer, you have 7 a.m. to 8 a.m. Maybe in case of winter, the time is 10 a.m. to 11 a.m. So you have a proper time to cut the root tip, and one of the precautionary method that has to be followed. And not only that, you have to see that the correct solutions are added at correct time, duration, and the fixative procedure. So all that has to be maintained, and during the heating process it should not be overheated, and these are the things that has to be kept in mind. And following that, the viva questions. So what are the possible viva questions that can be asked? So as I discussed in the initial stage itself, like the what is mitosis, meiosis, the stages. So in every practical experiment, I used to give an introduction, because that introduction are the information that relate with this experiment, and that gives you information about the viva, that gives you connect you with the viva question, possible viva questions that are asked in the practical examination. Hope we have discussed what is the mitosis that is employed in the onion root tip, how we take the tip of the onion root and subject it to different treatment and observe the different stages of mitosis of this onion root cell. Thank you.